Aqua-FAANG is a European Horizon 2020 project that aims to improve our understanding of fish biology to support our fisheries and aquaculture industries. It focuses on 6 important species that we use and farm within Europe. In this video, we will focus on the common carp. The common carp is also known as the European carp. It is an omnivorous freshwater species that prefers to live in ponds. Besides their highly commercial value, they are also very interesting for research. Due to its large size, different aspects of the carp can be looked at quite easily, such as the different organs or embryo development. In addition, the common carp is a species that underwent a whole genome duplication, which makes its genetics also interesting to look at. So about 12 million years ago, 2 other carp species that were swimming around within Europe decided to interbreed, and instead of both giving half their genetic material, they both gave their whole genetic material, which is quite unusual. This basically means that the common carp has 2 complete genomes instead of 1, thus a large amount of genes occur twice. The different types of data collected within Aqua-FAANG and their analysis will provide a clearer insight into the carp's genetics and epigenetics, and will give a better understanding of fish biology. In this video, we'll introduce 3 researchers of Aqua-FAANG who are working on the common carp, and how their research can help answer these important biological questions.
I'm Joanna Szygieł from Poland, and I'm researcher at the Institute of Ichthyology at Aquaculture in Gołąż, which is part of Polish Academy of Science.
In Aqua-FAANG, I'm involved in the work package 1, where using techniques like ATAC-seq we would like to generate functional annotation map for genome of common carp. These maps are a great tool to give us a view of genomic data of different fields of interest like gene function, basics of immunity, disease resistance, and so on. In ATAC-seq we are using an enzyme called transposase that is normally needed for transposition of DNA sequence that can change its position within a genome. This enzyme cuts accessible DNA, which is loosely wrapped around the nucleosomes, and thanks to that actively transcribed. Transposase leaves a gap for high-throughput sequencing adapters, which added to accessible DNA like kind of flags mark the regions of the genome with open chromatin, so we can get the information of chromatin organization and investigate the mechanisms controlling the work of genes. To reach this goal, we are working with different tissue types of fully developed carp, in a different way than Ada, who works with tiny, newly hatched fish.
Hi, I am Ana Jimenez Gonzalez, postdoctoral researcher in the University of Birmingham and collaborator with the Wageningen partners in the Aqua-FAANG consortium. I work with carp embryo samples in Work Package 1, and our main interest is to understand how the carp embryo can develop from a unique single cell to a more complex organism with tissues and organs. To answer this question, we have collected samples from carp embryos at different stages. In these samples, we can detect the molecular switches that turn on and off the genes coded in the embryonic DNA. These on and off marks determine how and when genes are read and translated into proteins, allowing the fish baby to grow up properly. This means that our results with mapping these marks will allow us to determine the conditions for a better fish health and development.
Hi, I'm Annemiek Blasweiler, PhD candidate at Aquaculture and Fisheries at Wageningen University here in the Netherlands. And I'm the last researcher in this video to tell you a bit about what we do with common carp within Aqua-FAANG. So within Aqua-FAANG I don't do any lab work like Ada and Joanna have so impressively shown you, but I'm a bioinformatician, or I'm trying to be. So basically what I do is take all the great functional data that they are collecting and really use it to look at immune genes within the common carp and how they function. But basically what I'm doing is looking at my DNA and RNA data from the common carp to see when the fish is undergoing an immune response, which genes are active and which are not, and which copy genes, so the sets of 2 genes, which copy is more active or are they active in the same way, and to see if they perform the same function. So with this I really get an overview on what is going on within the common carp when a virus or a bacteria invades, and this gives us powerful insights into what is actually happening within a fish when you have a lot of copied genes.